Did you know that scanning electron microscopes, commonly known as SEMs, can magnify objects up to 1 million times their original size? That's like turning a grain of sand into a boulder. It's a fascinating world that unfolds when you peer through the lens of an SEM. But how does it work? Pull up a chair because we're about to dive into the science behind this marvel of modern technology. At its core, a scanning electron microscope is a powerful tool that uses focused beams of high-energy electrons to generate a variety of signals at the surface of solid specimens. The term scanning comes from the fact that these electron beams scan the sample surface point by point, line by line, creating an image in a manner similar to how television pictures are produced. The process begins with the production of electrons. In the electron gun at the top of the microscope, electrons are boiled off a tungsten filament and accelerated towards the sample using a positive electrical field. This stream of electrons is then focused into a fine beam by a series of electromagnetic lenses. As this beam of electrons hits the specimen, it causes various interactions to occur. These interactions produce different types of signals, including secondary electrons, backscattered electrons, and X-rays. Each of these signals carries information about the sample's surface topography, composition, and other properties. Secondary electrons, for instance, are ejected from the sample as the incoming electron beam dislodges them. Because they come from the very surface of the sample, they provide detailed information about the surface topography and morphology. Backscattered electrons, on the other hand, are the original beam electrons that have been reflected back from the sample. They provide information about the contrast between different elements in the sample. X-rays, meanwhile, are generated when the electron beam knocks an electron out of an inner shell of a sample atom, causing an electron from an outer shell to drop down to fill the vacancy. This process emits energy in the form of an X-ray, which can be used to identify the elemental composition of the sample. The signals generated are then detected and processed to form an image on a computer screen. The brightness of each point in the image corresponds to the intensity of the signal received from each scanned point on the sample surface. This forms a detailed high-resolution image that provides a wealth of information about the sample. In summary, the scanning electron microscope is a powerful tool that uses a focused beam of high-energy electrons to probe the surfaces of solid specimens. It generates a variety of signals that provide detailed information about the sample's surface topography, composition, and other properties. The process starts with the production of electrons in the electron gun, followed by their acceleration, focusing, and interaction with the sample. The signals generated from these interactions are then detected, processed, and used to form an image. So the next time you look at an image magnified 1 million times its original size, remember the intricate dance of electrons, signals, and sensors that makes it all possible. It's not just a picture, it's a voyage into the unseen world made possible by the scanning electron microscope.